Good morning everyone. This is Dhandapani, qualified MSc B.Ed, Botany. I shall handle both Botany and Zoology coming from Coimbatore. So now let's have a small discussion about how a gene is regulated in prokaryotes. That is the lac operon concept. Lac operon concept. Actually, in molecular biology, there are three important topics which the teachers and the students feel difficult. These three topics are nothing but transcription, translation, and lac operon. These three topics, including one more topic, that is replication of DNA. That we can easily understand. So, replication of DNA, transcription, translation, and lac operon. These four topics are somewhat difficult or little bit difficult to understand. So among these four small topics, let's now discuss about the lac operon concept. It is actually how a gene is regulated in prokaryotes like bacteria. Prokaryotes, we already would have studied in the previous class, that is in the lower classes, about what is the prokaryotes and eukaryotes. The prokaryotic cells do not contain the original nucleus. It has a nuclear material that is called incipient nucleus, that is the DNA. The DNA acts as the nucleus, and it is not called the nucleus, it is nuclear material. It is not surrounded by the nuclear membrane, and membrane bound organelles like mitochondria, chloroplast, endoplasmic reticulum, Golgi apparatus, lysosome, ribosome, all the membrane bound organelles are totally absent in prokaryotes. In such prokaryotes, example bacteria and archaea, in such prokaryotes, how the gene is regulated. Now let's discuss. This is called lac operon concept. Okay, I will now explain what is lac operon. Here lac stands for lactose. Operon, what is meant by operon? Operon is a polycistronic structural gene. It is operon is a polycistronic structural gene. Actually, we know about the gene. So every human being is believed to have contain from 20,000 to 25,000 genes in our body. So human beings, we have 46 chromosomes, that is called in diploid condition, that exist in 23 pairs. So this 23 pairs of chromosomes totally contain 20 to 25,000 genes. So in prokaryotes, there is only one chromosome is present, that is in the bacteria, only one chromosome is present. So it contains a polycistronic structural gene. So here poly, we know that it means poly means many. Cistron, what is meant by cistron? It is a structural gene. It is a structural gene that codes for a polypeptide. Codes for a polypeptide or protein. Polypeptide or protein. Actually, all the characters from one generation to another generation, whether it is a prokaryotic cell or eukaryotic cell, whether it is a human being or any other lower organisms, the characters are transformed in the form of proteins only. So chromosomes contain DNA and proteins. So in bacteria, how the characters are expressed or how the characters are transferred from one generation to another generation. This is called lac operon. So lac operon means lac stands for lactose, operon stands for polycistronic structural gene. Now I draw the lac operon gene, polycistronic structural gene. This is T-O-I-P-Z-Y-A. This is a lac operon, polycistronic structural gene. Actually, in eukaryotes, only one gene, that is monocistronic structural gene, is responsible for transmission of characters. That is called transcription. Transcription means the process of copying the information from DNA into mRNA. The information that is available in the form of codes, genetic codes present in the chromosomes that are transferred into the single stranded mRNA. This process is called transcription. So it is monocistronic, only one cistron is found in the eukaryotes. But in prokaryotes, these 7 genes are present. That's why it is called a polycistronic structural gene. Here, P stands for promoter. In this polycistronic structural gene, here P stands for promoter, O stands for operator, and I is the regulatory gene. That is not called as an inducer, it is an inhibitor. I stands for inhibitor gene, inhibitor. Then P again stands for promoter gene. Then Z, Y and A, these 3 genes are called structural genes. Now let's explain about this expression of the gene regulation. Normally, when a bacteria are grown in the medium, that is in the culture medium in the laboratory, it needs a carbon source. So before studying this, let's study this lac operon concept under 2 headings. That is, in the absence of an inducer. So we are going to study the lac operon concept under 2 headings. First heading is in the absence of an inducer. Here, what is the inducer? Inducer is the carbon source. Inducer is the carbon source. Normally, when the bacteria are grown in the laboratory in the in vitro culture using MS medium or some other medium, it needs a carbon source. Normally, glucose is given as a carbon source because it contains carbon, C6H12O6. It contains carbon also. So normally glucose is given in the medium. Whenever glucose is given in the medium, the regulatory gene I produces a repressor mRNA. The inhibitory gene, that is regulatory gene. Actually, I stands for not inducer, it is an inhibitory gene. This inhibitory gene I produces an mRNA that is called repressor mRNA. This repressor mRNA produces a repressor protein. When this repressor mRNA and repressor protein are produced, only when glucose is added as a carbon source.